the prayer. So I'm going to begin the prayer. Okay, thank you everyone. Now, first day of the today's week 6 series is going to start with a, biology lecture. It will be delivered by Dr. Shivani Patel. She is Associate Professor and Head, Department of Biotechnology and Microbiology, RP University, Rajkot. She has vast experience in the field of molecular biology as well as genetics. She has experience in all the field, but overall, these are the area of interest where she is working and she has vast knowledge also. So without wasting time, I request Dr. Shivani Patel to start with the today's topic, gene interaction.
Good morning and Jai Samnarayan to everyone. I hope I'm audible to you.
Yes, ma'am.
Thank you for the response. So basically, as being shared with you, that the topic which we'll be going to share today is gene interaction. And I'm sure that because most of you are in your final year of bachelor's program, you would have gone through this topic at least once. If not now, then maybe a superficial introduction of this would have been covered in your school days when you were studying about Mendelian genetics. So this is a topic, from where you are likely to get almost 2 to 3 questions. May it be an exam for IISc or an exam of JNU, or even when you give CSIR net or GCET, always you find some questions from this gene interaction topic. So I thought I will just give you a brief overview and then we will go directly to the problem solving, just a revision session for most of you. And those who would have not covered, at least they will get some background which may help you to solve the problems in your examinations. So with this note, let us start. If I say genic interaction definition, so basically genic interaction is an expression of a character by the interaction of more than one pairs of gene. That is known as interaction of genes or genic interactions. So till now, when we have studied about Mendel's inheritance, we have seen that there are always two alleles which is responsible for handling a character. Say, for example, a height of a plant, right? So there was a scientist named Betson and Punnett. They have given an hypothesis which is known as factor hypothesis. And according to this hypothesis, basically, it says that this hypothesis, it says that, there are some characters which are being controlled by more than one pairs of gene. Just as TT is one pair of gene, which is controlling height of the plant, there would be one more. I mean, there are certain genes which are controlled by more than one pair of gene, right? By the interaction of more than one pair of the gene. This is one pair and this is another pair. So both of the pair interact to give you certain characters, right? So So this was being put forward by Bateson and Punnett, and they gave this factor hypothesis, which states that there are certain characters which are produced by the interaction of two or more pairs of the genes. Then these interactions basically are classified into two types. One is allelic gene interaction, another one is non-allelic gene interaction. So if I say allelic gene interactions, allelic gene interaction is an interaction between the two alleles which are located at the same locus of homologous chromosomes. I repeat again, the interaction between the two genes which gives the character, and these two genes are located at the same locus of homologous chromosomes. Then such type of interaction is known as allelic gene interactions. As you all know that we have homologous pairs of genes, homologous pair of chromosomes. So we have two chromosome number 1, we have two chromosome number 2. One we receive from mother parent, one set we receive from the father parent, right? So supposing I have got capital A gene from a father parent and a capital small a gene from the mother parent, my genotype is AA. And these genes are, supposing this gene is responsible for my eye color, then the gene responsible for the eye color in my father's chromosome, at the same position, the gene responsible for the eye color in my mother's chromosome will be present. That means these two are the genes which are located at the same locus of homologous chromosomes. And when the interaction between these two genes gives me some character, such type of interactions between the two alleles which are located at the same locus of homologous chromosomes is known as allelic gene interactions, because we all know that these are alleles. Alleles are the alternative forms of the genes which are located at the same locus of homologous chromosomes. Okay. Now, if I say non-allelic gene interactions. So basically, this allelic gene interactions are of three types. One is complete dominance, another one is incomplete dominance, and the third one is co-dominance. Okay. The second type of genic interaction is known as non-allelic gene interaction. The word itself tells you it is an interaction which expresses a character, but this interaction is between the genes which are non-allelic. That means this is an interaction which occurs between the genes that are located at the different locus of either the same chromosome or a different chromosome. So say, for example, this is chromosome number 1 and this is chromosome number 2. So supposing there is a gene A here, there is a gene B here, and there is a gene C here. Okay. So the interaction between the A and B gene to give a new character will be called as non-allelic gene interaction, because these are the genes which are located at the different locus of the same chromosomes, right? Or the interaction between A and C, if it is giving some character, then also it is a non-allelic gene interaction, because it is an interaction between the genes which are located at the different locus of different chromosomes, because this is located in chromosome number 1, the C is located in chromosome number 2. Okay, I hope you are very clear about this allelic and non-allelic gene interactions. I would just like to know, sir, is there any possibility that they respond to certain questions? Ravi, sir? Oh, can they respond to certain questions which I asked them?
Not yet, ma'am.
Okay, in the chat box, they can't respond.
Yeah.
Okay. So if I say the interaction between gene B and C will lead some character, whether this is a non-allelic gene interaction or an allelic gene interaction. Can anyone tell me? Yes, if you say it is a non-allelic gene interaction, you are correct, right? But if there is an interaction between B and B and it is giving you some character, whether it is an example of allelic gene interaction or a non-allelic gene interaction.
Allelic gene interaction.
Thank you. Oh, that's good. That means you are able to unmute yourself and speak. Thank you very much, whoever has responded. So I hope I'm very clear to you about these two types of interactions. We are going to solve the problems related to these two. So we should be very clear about these two terms. So There are basically 4 types of interactions which we will study under non-allelic gene interaction. Actually, there are many more, but the major and important ones I have tried to cover, that is complementary gene, supplementary gene, duplicative gene, and epistresis. Okay, so these are the 4 major type of interactions which are known as non-allelic gene interactions. So let us first see about allelic gene interactions. So these allelic gene interactions, as I have already told, that this is an interaction between the 2 genes that are located at the same locus of homologous chromosomes. And they are of 3 types: complete dominance. They show 3 types of it: complete dominance, incomplete dominance, and co-dominance. Okay. So if I say complete dominance, whatever Mendel has studied, he studied pea plant, and when he has crossed a purple color, whenever he has crossed a purple color flower plant with that of the white flower color plant, he found in F1 generation a heterozygous offspring in F1 generation, right? And all of them were purple. This is why, because see, this is allelic gene interaction. So it will occur between the genes located at the same locus. So whenever something occurs between the genes which are located at the same locus, we represent them with the same alphabet. So for recessive, we keep lowercase. For dominant, we keep uppercase, right? So here in this case, when 2 of them comes together, that is the lowercase as well as the uppercase alphabets comes together, this state is called as heterozygous condition, right? So in the heterozygous condition, usually one of the gene, this is purple color, the gene responsible for the purple color. So the gene, one of the gene completely masks the expression of the other. Right? It will completely mask the expression of the other, and that's why the whole F1 generation will be in purple color. Similarly, when you will self-cross the F1 generation, then we will find that in F2 generation, you will find 3 purple and you will find 1 white. Right? So white will be able to express only when both the alleles are of lowercase. So this type, what we have studied till now under Mendel's genetics, is actually the complete dominance what we have studied. So I think I don't have to explain you much. And you know that the phenotypic ratio for complete dominance will be, 3:1, and the genotypic ratio for the complete dominance will be 1:2:1. That is 1 capital P, 2 capital P, small p, and 1 small p, right? So this is, I hope, very clear to you. That is called as complete dominance. Another type of allelic gene interaction is incomplete dominance. It is seen in case of the flower color of Mirabilis jalapa, which is also known as 4 o'clock plant. So here, when you try to cross a red flower color plant with that of white flower color plant, in the F1 generation, you get a hybrid. That is, it will have 1 allele from the red flower color, 1 allele from the white flower color. So when they have a heterozygous condition, at that time, they will neither express red, neither completely express white. So the one gene is incompletely masking the expression of others. Right? So there is an incomplete expression by both the genes, and that is, it will give pink color, right? And when you will cross 2 pink color plants, then in F2 generations, you will find 1 red, 2 pink, and 1 white. That means their phenotypic expression, their phenotypic ratio, instead of 3:1 is 1:2:1. And even its genotypic ratio is 1 RR, 2 RW, and 1 WW. So here, in case of incomplete dominance, you see there is a change in the phenotypic ratio which was being given by Mendel as a monohybrid cross. The phenotypic ratio will always be 3:1. But in case of incomplete dominance, the phenotypic ratio will also be 1:2:1, and the genotypic ratio will also be 1:2:1. I hope I am clear to you. Why? Because one of the gene is not, both the genes are incompletely expressing itself, right? Or you may say that one gene cannot, the dominant gene is not completely able to express the recessive, completely mask the recessive gene. Okay, and that's why the genotype is 1:2:1. The genotypic as well as phenotypic ratio is 1:2:1. The third type of allelic gene interaction is codominance. And if we see codominance, both the alleles which are present at the same locus of homologous chromosomes, they both will express themselves completely when they will come together. So both of them are equally dominant on each other and no one can mask each other, right? So example for that is ABO blood group system. So we know that in the ABO blood group system, the A blood group has a genotype IA or IAIO, or sometimes we also write small I. B blood group has IBIB or IBIO. AB blood group is IAIB, and II, that is IOIO, will give O blood group type, right? Now here, whenever this A and B comes together, then both of them will express themselves completely, right? And that's why we have AB blood type. So this is an example of codominance. Another example for codominant is lentils coat pattern. Usually, lentils are the seeds, and on their surface of the seeds, either you get, these are white color seeds which has black spots in it. So either you found a spotted dentils, or this is the dotted dentils. Either you find the dotted dentils or you find the spotted dentils which has different shaped spots in it, right? So spotted dentils. Then there are marble dentils. There are so many types of lentils, but these lentils are actually the seeds which are white in color and they have black spots in it. So whenever you cross a spotted lentils with the dotted lentils, naturally a hybrid will be formed. And in the hybrid, you will find that when you will cross the dotted dentils with that of the spotted dentils, then in F1 generation you get a spotted dotted dentils. That means it will also have spots and it will also have dots. That means both of them are expressing themselves in the hybrid condition completely. So they are called as codominant, right? They are equally dominant and no one can mask each other, right? So that's why if A blood group male marries a B blood group females, and if they have an offspring, the offspring will always have AB blood type. That is, they have complete expression of mother's character as well as complete expression of the father's character in them. So codominance also, if you see, in the codominance, you see that both of them will completely express themselves. And that's why in the codominance also, in the F1 generation, whenever you will see its phenotypic as well as genotypic ratio, both the phenotypic as well as genotypic ratio will be 1:2:1 and 1:2:1. That is, the phenotypic ratio, instead of 3:1, they always have 1:2:1 because here both the characters are expressing themselves. So in incomplete and as well as codominance condition, the phenotypic ratio gets deviated from that of the Mendel's ratio which he proposed. Okay, so only complete dominant follows the Mendel's phenotypic ratio, that is 3:1 for monohybrid cross, right? So in case of, in complete dominance, you will find 3:1, whereas in incomplete dominance as well as in codominance, you will find that the phenotypic ratio will be 1:2:1. So either if they incompletely express themselves when they are together, or they will completely express themselves when they are together. So I hope it is clear to you. Let us take a problem related to Mendel's, related to the allelic gene interactions. So the question is here. I wish you, those who read this, try to solve it based on whatever discussion we have done or prior knowledge which you have, and unmute yourself and speak right answer, whether it is A, B, C, or D. So the question is, if a man of blood group AB marries a woman of blood group A, whose father was blood group O, to what different blood groups can this man and woman expect their children to belong to? Yes, anyone can tell the right answer? Okay, I go for it. So supposing a man is there, they say. Okay, some of them are responding C as been told. So if I say it is not C, because you see, they have told the male which is there, male has AB blood group. That means it has IB. And the female, if you see, she has A blood group. So A blood group could be either IA or it could be IAIO. Now, it has been told to us that the woman's father was O blood group. O blood group means it should have IOI. Could be also written as small I, small I. Okay. So now, when she will definitely receive one allele from the father parent, because this woman, she has a blood group. She has a blood group A. So her father is O. So her father is O means she will definitely receive one allele from the father. So there should be IO. So when it is IAIO crossed, then you see that you have IA, you have IAIO, you also have IAIB, as well as you have IBIO. So you have A blood group, you also have AB blood group, and you also have B blood group. Right? So the right answer is A. Okay, thank you. So the right answer is A. Now going to the second question. The second question is, in a human blood system, ABO blood system, the allele A and B are dominant to O. What will be the number of different possible genotypes? So you have A, B, and O. Alleles are there. These are the three multiple alleles responding for a single character, that is blood group, right? So what are the different possible genotypes? IA, IB, and IO are there. What are the different possible genotype? How many number? So if they're responding, please let me know. I can't see their screen.
Ma'am, C.
Very good. 6, right? C. So how it is C? Because it will have IAIA. It may also have IAIO. It will have IB, or it will have the blood group B may also have IBIO. You have blood group AB, which is IAIB, and you have O blood group, which is IOIO. So total is 1, 2, 3, 4, 5, and 6. Right? So this type of question comes actually from the allelic gene interactions, which I feel is very simple for you. But let us go to the non-allelic gene interactions. The questions from this non-allelic gene interactions, if you see, are more complicated and requires more time to solve. But if you are clear with certain tips, I think it is very easy for us to solve these questions with confidence in a short period of time. So as I told, non-allelic gene interactions are the interactions between the two genes which are located at the same locus of, which are located at the different locus of the same chromosome or different chromosomes, right? Those are called as non-allelic gene interactions. So in non-allelic gene interactions, instead of single alphabet, you will definitely have two alphabets, right? There will be two different pairs of the genes which are involving themselves, which are interacting with each other to give you a character. Now, let us first of all see what are complementary genes. So, complementary genes are two or more non-allelic dominant genes which interact with one another to produce a character. But one gene cannot produce the character in the absence of other. Okay, so these are two independent genes, but they will interact to give you a character. But to express that character, both of them needs to be in the dominant condition. Then only they will express themselves. Okay. So example is a flower color in the P. Example is flower color in the P. So say for example, there are two genes, that is gene C and there is a gene P. Okay. So gene C, which is there, is responsible to convert the precursor into an intermediate product. And gene P is responsible to convert this intermediate product into the pigment, that is anthocyanin, which is purple color, right? Now, if you have small c, then what will happen? The precursor will not get converted into intermediate product. So only capital C will not be able to produce anthocyanin, and only capital P will also not able to. Supposing there is a capital P and there is a small c, then what will happen is precursor will not get converted into intermediate product. And so even there is an enzyme which is being produced by this P gene, then it will not be able to convert into anthocyanin. So to make anthocyanin or to make purple color plant, you require both of them in a dominant conditions. Okay, so you require both of them in the dominant conditions. Okay, so so this type of they are codependent on each other. That means there is a gene A. Supposing there is gene 1, which is AA, and there is gene B, that is gene 2, which is BB. So these both, to express themselves, both are codependent on each other. That means both of them should express, both of them should be in the capital form, dominant form, to express the character, right? And that's why, you know, the ratio when you go for, I hope all of you know about the dihybrid cross ratio. What is the phenotypic ratio for a dihybrid cross given by Mendel? Phenotypic ratio?
9:3:3:1.
Very good. It is 9:3:3:1. But because of the complementarity, because they are interdependent to express a character, this ratio gets changed. The ratio gets changed too, because you will find, you know, according to the Mendel, when you go for a dihybrid cross, you get 9 capital C, capital P, 9 which is capital C, small, 3 which is capital C, small p. There'll be 3 which is capital P, small c, and there will be 1 which is small c, small p, right? So 9:3:3:1. Now in this case, we know that this all 9 will be colored. That is, they will be able to produce a purple color flower, right? But these 3 will not be able, because here capital C is there. So intermediate product will form, but anthocyanin can't be formed. Here, you know, the intermediate product is not forming, and so anthocyanin cannot be formed. And in this case, none of them are expressing. So all these 3, that is 3 plus 3 plus 1, that is 7 of them will be white, and 9 will be purple. So here, whenever you go for a dihybrid cross for the complementary genes, instead of 9:3:3:1 ratio, usually you get 9 purple is to 7 white. So here the ratio comes out to be, for the complementary gene action, the ratio comes out to be 9:7. Why I have kept this interaction as the first? Because I have seen, when I have analyzed different types of questions from different types of exams, maximum question comes from this complementary gene interactions. Maximum questions, I will give you those questions. In a different form, they try to ask questions from this complementary gene interactions. Okay, so we should understand that the complementary gene interaction is 9:7. Some books, you know, mentions this complementary gene interaction. The word complementary itself tells you that both of them are required. They're complementary to each other. Day and night, they're complementary to each other. You cannot define one in the absence of other, right? So these are complementary. But another name for this complementary gene expression is called as double recessive. It is also called as double recessive. It is also called as double recessive epistasis. In some books, they mention it as double recessive epistasis. I will come to this later on, but I wanted you to understand, with the words itself, you can understand complementary means both the genes needs to be in the dominant form to express themselves, right? That is known as complementary gene interaction to give a character. The second type of gene is supplementary gene. So these are also 2 independent dominant genes which can individually produce. Here also there are 2 genes, that is, supposing one gene is AA, another gene is B. So both of genes actually can express themselves. Both of them can, they are not codependent. They are not dependent on each other. Both of them can express themselves. But you know, when they are present together, they give altogether a new type of character. When they are present together, they give a new character. So if I read this, supplementary genes may be defined as 2 independent pairs of dominant gene which interact in such a way that each dominant gene produces its effect whether the other is present or not. But when the second dominant gene is added to the first, a new character is expressed. So the example is combs in the fowl. So usually combs in the fowl are of three types. One is single, you can see this type, walnut, pea and rose. Okay, these are the four types of combs which are there. So this rose comb is because of capital R gene, P is because of the small p gene, and whenever you have, it is because of capital P gene, this is capital B gene, this is capital R gene. When both of them are recessive, that time it will produce, when both of them are recessive, they will produce single type, and when both of them are dominant, that is when both of them are present together, they produces altogether a new type which is known as walnut. And that's why whenever you do a cross, I think I now don't have to repeat, the dihybrid cross will always have 9 capital R capital P, there will be 3 which is capital R small p, there will be 3 again which is having small r capital P, and there will be 1 which is small r small p. So 9:3:3:1. I already told that capital R gives you rose, right? And when both of them are together, they give all together a new look which is known as walnut, right? And when P is there, it will give P. And when both of them are recessive, it will give single. So here you find 9:3:3:1 ratio. But here this 9 is, a character which is a new character added by the contribution of both the genes. Supplementary. Each of them supplements to give something new. Just as in your exams, you write supplementary which adds on to whatever you have written to the main sheets, right? So supplementary, 2 of them when they are together, they give something new. And so they will be giving you 9 walnut is to 3 rose is to 3 pea is to 1 single. Okay, so 9:3:3:1 ratio is there for the supplementary gene. Only thing is 9 is actually a new character which is added, which was not present in either of the parents. Okay, is it clear? May I move forward?
Yes, ma'am.
Thank you.
Yes, ma'am.
Thank you very much. So the third type of non-allelic gene interaction is duplicative genes. So when I say duplicative genes, again, they are the genes. Say for example, AA and BB, these are the 2 different genes, right? 2 different genes. Both of them, actually, there is supposing X, which is making some character, right? So both A and B, right? A or B, both of them will be helping us. The product of B may also convert X into character, and the product of A also may convert the X into a character. Okay, so any of them, the presence of either A or either B, both of them will express the same character. Okay, so if both of them are present, then also that character will be produced. Either of one is present, then also that character is produced. Right? So when a single character is controlled by 2 or more pairs of non-allelic independent genes, then it is called as duplicative gene. It is also a non-allelic gene interaction. So here you can see this is a precursor. It is producing a product. So the product of A gene will also result in converting precursor into product, and the product of B will also result in converting precursor into product. Right? So the example is triangular seed case in certain wheats. So here the capital T gene is also responsible for the triangular seed shape, and capital V is also responsible for triangular seed shape, right? So in F2 generation, if you see the ratio, always the ratio will be, the ratio which you will get in the dihybrid cross for the F2 generation will be capital T, capital V. It is capital T, small v. It will be small t, capital V, or it will be small t, small v. So in this case, all this will also give a triangular shape. This will also give triangular shape. This will also give triangular shape. So 9 plus 3 plus 3, that goes to 15. So you will have 15 triangular is to 1 oval. Right? This is called as 15 triangular is to 1 oval. So the ratio here, instead of 9:3:3:1, you have 15:1 ratio. Why? Because both the genes are able to express the same character. Is it okay? So these duplicative genes which are there, they are also known as double dominant. The duplicative genes are also known as double dominant genes. They represent double dominant epistasis. So duplicative, I have said, is both of them are performing the same function, right? So whether they are present alone or when they are present together, same functions they will be presenting. Okay, the ratio for duplicative gene is 15:1. Coming to epistasis, which is the most important chapter of this non-allelic gene interactions, and many questions from this epistasis come. Even what I have told as complementary gene or duplicative gene, the questions related to that also comes under the heading of epistasis. Okay, so if I say epistasis, the literal meaning of epistasis is to stop or to mask or to inhibit. So epistasis is the prevention of expression of a gene. So if there are two non-allelic genes, one gene will actually mask the expression of other. It will suppress the expression of others, right? So this, one gene will not allow the other to express, or it will inhibit the expression of other, or will prevent the expression of other. Then that phenomenon is known as epistasis. And the gene which is inhibiting, in this case, A is inhibiting B. So the gene which is inhibiting is known as epistatic gene, and the gene whose expression gets inhibited is known as hypostatic gene. Okay, and this epistasis could be majorly classified into two types: dominant epistasis and recessive epistasis. Dominant epistasis is when the prevention of expression of gene is by a dominant gene, then it is dominant epistasis. When the prevention of expression of a character is by a recessive gene, it is called as recessive epistasis. Okay, so if I say dominant epistasis, so this I don't think needs to be explained. If I say dominant epistasis, then the prevention of expression of a gene by a dominant non-allelic gene is known as dominant epistasis. Example is in the case of leghorns. In the case of leghorns, there are two types of varieties. The one is the colored birds, and another one is the white birds. Right? Two types of varieties are there. The colored nature is because of capital C. It is because of a capital C gene. But this capital C gene, if there is another gene which is in the dominant form, that is I gene, then it inhibits the expression of the C. So the C and I, if there together, it will not be able to give a colored character, but it will be able to give no color, that is white. Why? Because this capital I inhibits the C, right? But when you have capital C, or capital C when it is there, and with that if you have small I, then only it will give you colored birds. Okay, this is why, because capital I inhibits the expression of capital C. Small I cannot inhibit the expression of the capital C. And if there is small c, then by default it will be white color. So here in this case, when we cross white leghorns, this is white. Why? Because this is genetically colored. Sorry, it is genetically colored because it has capital C. And when it is crossed with a small c, with this small c itself will not give you any color. In the F1 generation, instead of colored character, it gives us white character. Why? Because this capital I is inhibiting capital C. So here in this case, when you find 9:3:3:1 ratio, with the capital CI, it will give white color. With the capital C and small I, it will give colored, right? But when you will have a small c with capital I, then also it will give you white. And when it is small c, small I, then also it will give you white. So if you see, how many white are there? 9 + 3 + 1, right? So you have 13. You have 13 which are white, and you have 3 which are colored. This is called as, so for the dominant epistasis, the ratio is 13:3 instead of 9:3:3:1. Okay, so we have seen complementary genes has 9:2. We have seen that the complementary colors have 9:3:2. We will just go again to another, that is recessive epistasis, and then we will revise all of them, right? Now, just as dominant epistasis has 13:3 ratio, because when a dominant non-allelic gene prevents the expression of another gene, then it is known as dominant epistasis. Similarly comes as recessive epistasis. So recessive epistasis, the prevention of expression of a particular gene by a recessive non-allelic gene will be called as recessive epistasis. The example for this is coat color in mice, right? Coat color in mice. So there are basically three types of coat color. One is agouti, which is like gray, then black and albino, right? Now agouti color is produced. Agouti color is produced when you have capital M and capital A, right? They produces agouti color. Capital M actually produces black color. Capital M produces black color, right? And capital A, which is there, supposing it produces black color. When capital A is there and you have small m, then the expression of capital A is prevented by small m. So this is not able to produce any color, right? And so small m will give you albino. No color will be produced. Why? Because this capital A is producing some color because of which you get a gray color. But in the presence of the recessive set of small m gene, it is not able to express itself, right? So the recessive non-allelic gene is inhibiting the expression of A. So you get albino, right? So here, when you see 9:3:3:1 ratio, what happens is 9 you have M and A, you have 3 which is M and small a, you have 3 which is small m, capital A, and you have 1 which is small m, small a, small a, right? So here, this is going to give you agouti, right? This is going to give you agouti. This is going to give you black. But this is not able to express its color in the presence of a recessive set of non-allelic gene, and so it is white. So it is white, and this is already white. So here the ratio comes, this 2 white gets plus, so this ratio comes as 9:3:4, right? So the ratio for a recessive epistasis is 9:3:4. Okay, so this is why, because just as in a metabolic pathway, there are a lot of intermediate products which form, and then you find a final color, right? Now here, what happens is, although you have capital A here, but if this is small m, then what will happen? This metabolite 1 will not be able to convert into 2. So although you have capital A gene, right, the recessive form of this gene is actually inhibiting the expression of this gene, and you are not able to show melanin production, right? So this is the recessive set of non-allelic gene is preventing the expression of a gene, right? Now here I would like to tell that, these are the tip. This is the most important slide. I think you should keep it in your mind, and I also wish you to write it in your book if you have with you. So, this epistasis, the whole problem related to, all the problems related to this non-allelic gene interaction which comes revolves around this ratios only. So how to remember these ratios? I hope all of you will remember dihybrid cross ratio, because since from our school days we are reading about dihybrid Mendel cross phenotypic ratio is 9:3:3:1, right? Now dominant this is dominant epistasis, okay? I'm sorry, this is dominant epistasis, okay? So dominant epistasis, the ratio is 12:3:1. Abhi humne dekha tha, dominant epistasis mein, we have seen the example of leghorn's color, that is colored and white type of wing, and C and I gene, both of them, the capital I gene is inhibiting the expression of the small I. So we have got 12:3:1 ratio, right? So कैसे याद रखना है आपको? जैसे डाइहाइब्रिड क्रॉस है, उसका रेशियो होता है 9:3:3:1, है ना? उसका रेशियो होता है 9:3:3:1. तो आपको कैसे याद रखना है? तो इसके लिए आपको इस तरह से याद रखना है कि जो डोमिनेंट एपिस्टेसिस होता है, उसको इस तरफ से आप 2 को प्लस कर दोगे, तो 9 + 3 कितना होता है? ये डोमिनेंट साइड है, ये वाला डोमिनेंट साइड है, ये रिसेसिव साइड है ना? तो जब डोमिनेंट साइड से प्लस करोगे तो उसका रेशियो आता है 12:3:1. ये दोनों को प्लस कर दोगे, राइट? रेसेसिव एपिस्टेसिस के लिए आप इस तरफ से प्लस करोगे, इस तरफ से जाओगे, जो नॉर्मल डाइहाइब्रिड क्रॉस है उसको इस तरफ से प्लस करोगे तो कितना होगा 9:3:2, ये दोनों को प्लस करोगे तो कितना होगा 4, राइट. सो डोमिनेंट कैसे याद रखना है कि इन लोग का रेशियोस क्या रहता है वो बता रही हूँ कि डोमिनेंट एपिस्टेसिस के लिए आप इस तरफ से प्लस करेंगे, रेसेसिव के लिए आप इस तरफ से प्लस करेंगे. जो डुप्लीकेटिव जीन है उसको हम लोग डबल डोमिनेंट एपिस्टेसिस भी बोलते हैं. सो डबल डोमिनेंट एपिस्टेसिस में क्या करेंगे, आप ये दोनों को लेंगे, उसके साथ एक और भी लेंगे. सो 9 + 3 12 + 3 कितना होता है 15, तो हो जाएगा 15:1. सो डुप्लीकेटिव जीन का रेशियो आता है 15:1 और इसलिए उसको डबल डोमिनेंट एपिस्टेसिस भी बोलते हैं. ठीक है. और जो कॉम्प्लीमेंट्री जीन है उसको हम लोग डबल रिसेसिव बोलते हैं. डबल रिसेसिव मतलब आप किस तरफ से, राइट साइड से प्लस करोगे कि लेफ्ट से करोगे, इधर से करोगे, राइट. यू विल गो फ्रॉम दिस साइड टू दिस साइड. सो इट विल बी 9:7. To be very frank, most of the questions, whichever I have seen till date, maximum question from non-allelic gene interaction comes from this complementary gene in number of different ways. Okay, and there is another type of dominant and recessive epistasis. If we see some questions related to that, उसमें हम लोग 9 + 3 + एक 3 को छोड़ देते हैं और 1. So that goes to 9 + 3 + 1 हो गया 12 and 13. So हो गया 13:3. Right, it will go to 13:3 is the ratio. So in fact, you should always remember that if you remember a dihybrid cross ratio, then it is very easy for us to summarize. It is very easy for us to summarize that this is a ratio for a dihybrid cross, right? This is a ratio for a duplicate recessive. This is a ratio for a recessive, because recessive में हम लोग क्या करेंगे, इस तरफ से 2 को प्लस करेंगे तो 4 आ गया, right? फिर डोमिनेंट एपिस्टेसिस हम लोग इस तरफ से 2 को प्लस करेंगे, मतलब आ गया 12, राइट. सो 12:3:1 आएगा. और भी एक डोमिनेंट एपिस्टेसिस होता है जिसमें मैंने बताया कि हम ये 2 को और इसको प्लस करते हैं, जिसको इसको हम लोग डोमिनेंट एंड रेसेसिव एपिस्टेसिस भी बोलते हैं, राइट. इसको हम लोग डोमिनेंट एंड रेसेसिव एपिस्टेसिस भी बोलते हैं, जो 13:3 आता है. और एक डुप्लीकेट डोमिनेंट होता है जो 15:1 आता है, क्योंकि डुप्लीकेट डोमिनेंट में आप यहाँ से 3 को प्लस करेंगे. सो 9 + 3 + 3 15:1. सो इस तरह से हमको याद रखना रहता है. ये एपिस्टेसिस, ये जो डिफरेंट टाइप के नॉन एलिक जीन इंटरेक्शंस हैं, वो इस तरह से हम रिलेट कर सकते हैं और इस तरह से हम उसको पढ़ सकते हैं, राइट. लेट अस गो टू प्रॉब्लम सॉल्विंग. वी हैव हाउ मच टाइम सर?
5 to 10.
Oh. Okay, let us go fast. Okay. In case of two gene interaction, the gene which is masking the expression of another is called, and the gene whose expression is masked is called. Please tell me 1, 2, 3, or 4. Fast.
3.
Very good.
C.
Oh,
3.
very good. Thank you. So it is a correct answer is 3. Okay, that is the one which is masking the expression is epistatic, and the gene whose expression gets masked is hypostatic. Second question is, what is the dihybrid phenotypic ratio of a recessive epistasis is in F2 generation? Jaldi se. Recessive
9:3:4.
D.
से आप प्लस करेंगे, राइट. सो 9:3:4. recessive epistasis में इस तरफ से प्लस करेंगे. गुड. So answer is D. Next question is, following is the hypothetical biochemical pathway responsible for pigmentation of leaf. The pathway is controlled by 2 independent assorting gene A and B encoding the enzyme as shown below. Okay, so A is producing yellow color, B is producing green color, and when both A and B is not there, it will be white color. So if you see what is expected progeny after selfing a plant with the genotype AABB. So when you self AABB, you will get a dihybrid cross. Okay, when both of them are there, which color you will get? When A and B are there, then it will give you green color, right? When only A is there, you will get yellow color. When only B is there, you will get no color, that is white. Oh, sorry, you will get yellow color again, sorry. When B is small, then it is stuck here itself. And when you have both this white, so you get 9 green, you get how many yellow? 6 yellow and 1 white. So ओके, ये कुछ गलत हुआ। हैं?
मैम जो एक ये बी है सिर्फ उसमें वाइट ही आएगा ना।
हाँ वाइट आएगा, वेरी ट्रू, क्योंकि ए येलो बना ही नहीं रहा है, राइट। थैंक यू। दिस इज ए, सो दिस विल बी वाइट। व्हाई इट इज वाइट? क्योंकि ए ही नहीं है तो ये वाई बनेगा ही नहीं, राइट। सो ये वाइट रहेगा, तो इट विल बी 9:3:4. So 9 green is to 4 white is to 3 yellow, right? So which will be the right answer? The first one is the right answer. Thank you. May I know the name of a girl who is responding? What's your
Ishita.
Ishita. Good. Which college you belong to?
PDPS चारू से।
Very good. Very good. Now this is the next question to you. Two mutant plants, both bearing white flowers, were crossed. All F1 plants has red flower color. F1 plant has self to produce progeny with either red or white color in the ratio 9:7. Whenever 9:7 ratio comes, means what it is?
Double recessive, complementary.
Very good. Very good. So I said most of the question comes from this, right? अभी तो रेशियो ही दे दिया है, तो सीधा ही हम ये पढ़ सकते हैं। तो पहला पूछा है म्यूटेशन इन द पेरेंट्स डू नॉट कॉम्प्लीमेंट ईच अदर, तो ऐसा तो है नहीं, दोनों कॉम्प्लीमेंट्री है ना। आपने बताया इट इज अ डबल रिसेसिव, तो ये गलत आंसर है। म्यूटेशन इन द पेरेंट्स आर एलेलिक, ये भी गलत है। म्यूटेशन इन द पेरेंट्स आर नॉन एलेलिक, ये शायद सही है। चौथा है द म्यूटेशन इन द पेरेंट्स आर लिंक्ड, ये भी गलत है। तो राइट आंसर क्या होगा? द म्यूटेशन इन द पेरेंट्स आर नॉन एलेलिक। अब यहाँ आप ध्यान से एक बात मेरी सुन लो, कई बार क्या होता है वो लोग ये रेशियो नहीं देते, कई बार वो लोग ये रेशियो नहीं देते हैं, और ये रेशियो ना देके वो लोग यही बोलते हैं कि जब दो वाइट म्यूटेंट प्लांट को हम लोग क्रॉस करते हैं तो वो एक रेड प्लांट देता है, राइट। और अगर उन्होंने ऐसा दिया हो, रेशियो ना दिया हो, और आपको यहाँ पे ऐसा दिया हो, दैट इज द म्यूटेशन इन द पेरेंट्स कॉम्प्लीमेंट ईच अदर, तो फिर क्या हो जाएगा? जब भी कोई दो वाइट रेड देता है तो हमेशा वो कॉम्प्लीमेंट्री जीन ही होता है, राइट। क्योंकि दोनों सेल्फ एक्सप्रेसिव नहीं थे, पर वो लोग को डिपेंडेंट थे। जब साथ में आ गए तब वो लोग ने कलर एक्सप्रेशन करना शुरू किया। तो वो केसेस में ये दोनों राइट आंसर होंगे जब रेशियो नहीं दिए जाते हैं, राइट। तब कई बार सीधा पूछते हैं दो वाइट रेड को प्रोड्यूस करता है, ठीक है। सो इसमें राइट आंसर होगा नंबर 3, दैट इज द म्यूटेशन द पेरेंट्स आर नॉन एलेलिक। फिर आता है अगेन, देखो ये अलग तरह से फिर से कॉम्प्लीमेंट्री जीन्स के बारे में पूछा है। द जेनेटिक टेस्ट 9:3 रेशियो इन F2 जनरेशन रिप्रेजेंट्स व्हाट? एपिस्टेसिस। वेरी गुड, राइट। सो इस तरह से भी वो पूछता है। नेक्स्ट इज कोट कलर ऑफ डॉग डिपेंड्स अपॉन द एक्शन ऑफ एटलीस्ट 2 जीन्स। वन लोकस इज डोमिनेंट एपिस्टेटिक। देखो अभी पहले ही आपको नाम मिल गया है। Inhibitor of the coat color pigment A prevents the expression of color alleles at another independently assorted locus, producing white color coat color. When the recessive condition exists at the inhibitor locus, the alleles of the hypostatic locus may be expressed. So small a b produce black color, and small a small b produce brown color. When the dihybrid white dogs are mated together, determine the phenotypic proportion expected in the progeny. So
12:3:1.
Right, it is a dominant epistasis, means यहाँ से सीधा प्लस करके मुझे 12:3:1 लिख देना है। दूसरा, क्योंकि उन्होंने बोला है कि स्मॉल ए स्मॉल बी भी ब्राउन कलर देता है, तो इसमें किसी में भी आप देखें कहीं पे भी 1 आता ही नहीं, हमेशा ये 1 ही रहता है ना। सो यहाँ पे कहीं पे भी बाकी 3 ऑप्शन में 1 आया नहीं है, तो भी आप डायरेक्टली उसको सिलेक्ट कर सकते हैं, या अगर आप पहले ही लाइन में पकड़ लेते हैं तो भी आपको पता है, तुरंत आप कैलकुलेट करके यू कैन राइट 12:3:1. सो द राइट आंसर इज सी। नेक्स्ट क्वेश्चन, सेम क्वेश्चन हैज बीन आस्क्ड इन अ डिफरेंट वे। मैंने बताया था दो वाइट फ्लावर कलर को क्रॉस करोगे और अगर रेड मिलेगा तो ये क्या, इन व्हाट रेशियो विल द रेड एंड व्हाइट अकर? ये भी कॉम्प्लीमेंट्री जीन का, यस, ये भी कॉम्प्लीमेंट्री जीन का एग्जांपल है। तो मैंने बोला आपको मैक्सिमम क्वेश्चन रेसेसिव, डबल रेसेसिव से ही आते हैं, दैट इज अलग अलग तरह से उसको पूछा जाता है, राइट। नेक्स्ट क्वेश्चन, व्हेन द 2 independent pure lines of P with white flower colors are crossed, the F1 progeny was purple. the F2 progeny produced on selfing again has showed what? 9:7. The following conclusions were made. First is 2 different genes are involved, mutation in which lead to the formation of white color. ये सही लगता है? Yes, है ना? The 2 genes shows independent assortment. Yes, वो लोग 2 different gene हैं तो independently assort होंगे. This is an example of complementary gene interaction. 9:7 itself tells you it is a complementary gene interaction, and it is an example of a duplicative gene. ये गलत. तो इसमें right answer क्या होगा?
Fourth.
Perfect. आप लोग सॉल्व कर लेंगे. आपको जो मैंने टिप सिखाई है, जो आपको एक ही वो दिखाया था, उसी से आपको सब कुछ सॉल्व करना है, राइट? टिप्स जिसमें आपको डबल डोमिनेंट एपिस्टेसिस, रिसेसिव एपिस्टेसिस, डबल डोमिनेंट एपिस्टेसिस, डबल रिसेसिव एपिस्टेसिस एंड डोमिनेंट एंड रिसेसिव एपिस्टेसिस. इस तरह से अगर उसको याद रखेंगे तो आप कोई भी प्रॉब्लम सॉल्व कर सकते हैं, राइट? इन द फॉलोइंग एग्जांपल 3 independently assorting genes are known to govern a coat color in mice. The genotype of few coat colors are given below. What is expected? What is expected frequency of albino in the F2 progeny cross when blue back to the albino in the genotype AABBCC? Pure black with albino. Pure black with albino. Pure means it is like this only. With AABBCC. Anyone got the answer? Will come to 16/64. So that comes to 1/4. So the right answer is 1/4. Okay. I think we are running short of time, but these questions would be with you. Please go through it. This is the last question which I want you to attempt. A hypothetical biochemical pathway for the formation of eye color in the insects is given below. Okay, an autosomal mutant A and B. That means if you have capital A, it will give brown color. If you have capital B, you have orange color. And if both of them are present, it is orange-brown. So it is an example of a co-dominant expression. So consider the mutants are not linked. What is the phenotype of F2 progeny when you are crossing AABB with AABB? When you are crossing AABB with AABB. So FN progeny is intercrossed, then it will be AABB. And if you are intercrossing it, that means it will give you 9:3:3:1 ratio. I think when you cross this, 9 capital A, capital B, capital A, small b, small a, capital B, and small a, small b. So when you have both of them, you get orange brown, right? When you have only A and no B, you will get orange. No, sorry, you get brown. I'm sorry. And if you have only B, no A, then you will have orange. And when you don't have anything, it will be colorless. So which is the right answer here? 9 orange brown, 3 brown, 3 orange, and 1 color. So I think first is the right answer, right? So this is the right answer. So thank you all for your valuable time. I hope some tips shared to you here in this lecture would be useful. Wish you all very best. Thank you.
Thank you so much, ma'am. These are very important, actually, topics, and the way by which you explain, it is really amazing. And these questions will be with students only. Ma'am will send her PPT, and
Yeah,
that
sure.
will be available to students.
Sure, sure.
Thank you.
Thank you, everyone.
Now, next lecture will going to begin with. It is based on zoology. The topic is nervous system and impulse transfer. This topic will be delivered by one of the very dynamic faculty, Dr. Patel. He is assistant professor in zoology department, Government Science College, Limkheda. He has great experience in the field of competitive exam preparation. So I'm just requesting Sir Patel sir to begin with his lecture.
Thank you, Ravi sir. Am I audible?
Yes, sir, you are audible. Perfect.
Okay, sir. Can I share my screen?
Yes, ma'am. Yes, sir. Yes, share your screen.
Okay, sir.
Yes, sir, it's visible.
सबको दिख रहा है सर?
Yes, sir, बिल्कुल दिख रहा है.
Okay. Welcome all students. आज हम देखने जा रहे हैं नर्वस सिस्टम पे. हम लोग काफी टाइम पे देखते हैं कि दिमाग को दौड़ाना पड़ता है. आज थोड़ा दौड़ाएंगे भी, सही में. ठीक है. नर्वस सिस्टम तो हम शुरू करते हैं सबसे पहले कि नर्वस सिस्टम होता क्या है. देखिए, हमारा सेशन थोड़ा हिंदी एंड इंग्लिश में रहेगा. ठीक है, तो क्या कुछ गुजराती मीडियम वाले, इंग्लिश मीडियम वाले सबको भी कंफर्टेबल रहे. तो नर्वस सिस्टम सबसे पहले देखते हैं नर्वस सिस्टम होता क्या है. तो इट इज अ सिस्टम विच इज कंपोज्ड ऑफ ब्रेन, स्पाइनल कॉर्ड एंड द नर्व्स ओरिजिनेटिंग फ्रॉम दीज टू ऑर्गन्स. जनरली इसमें सेंटर में दो ऑर्गन रहते हैं, एक ब्रेन होता है, एक स्पाइनल कॉर्ड होता है. एंड उस ब्रेन में से 12 पेयर क्रेनियल निकलती है एंड स्पाइनल कॉर्ड में से 31 पेयर